In this video, I'll be covering developmental networks. A developmental network is a series of enzymatic interactions that can lead to a phenotype. And in this case, we have thorny dragons, and their phenotype is spikes. Here are several examples about how developmental networks can be used to show the phenotype being expressed or not. And so here we have the wild type strain, so all DNA is unmutated. So we have the S enzyme will promote P. P inhibits I can no longer inhibit K, so K is allowed to promote E, and then E promotes the formation of spikes in the skin cells. Secondly, here we have a mutated strain of DNA, so we have a P mutation where the P is no longer functional. So in this situation, we have S will allow the promotion of P, but since P is no longer functional, it cannot inhibit I, and so I is allowed to inhibit K, which will no longer promote E, and E will no longer promote the formation of spikes. So in this case, no spikes will be formed in the skin cells of the thorny dragons. Thirdly, here we have an I- mutation where the I enzyme is no longer functional. So we have S will promote P, and P will inhibit I, but since I is already non-functional, it doesn't really matter. It's kind of redundant. So K is allowed to promote E, and E promotes spikes. So spikes will be formed in this scenario. Fourth, here we have an I- and E- mutation, so the I and E enzymes are no longer functional, and so in this scenario we have S promotes P, which inhibits the already non-functional I, and so K is allowed to promote E. The E-, however, does not promote spikes because it is not functional, and so in this scenario no spikes will be formed in the skin cells. And lastly, here we have an S- and K- mutation. So in this scenario, the S will no longer promote P since it's S-, and so P cannot inhibit I, so I is allowed to inhibit the already non-functional K, and so in this case, no E will be formed because the K cannot promote it, and so therefore no spikes will be formed in the skin cells because of this mutation. Now that we know about developmental networks, we can approach this question. So why don't all skin cells in thorny dragons produce spikes when they have the same DNA? So the answer to this is epigenetics, and I will explain more about this. Epigenetics is the heritable changes in gene expression that occur without changing the DNA structure, and it involves the regulation of the gene products themselves. If the gene products of one of the skin cells of a thorny dragon are inhibited by something other than a DNA sequence change, then the spikes that would have been expressed are no longer expressed in the cell. And so a process that could cause this would be DNA methylation, in which the DNA is flagged with methyl groups as a way to tell the cell not to produce any of the gene products of this sequence of DNA. And so methylation, along with other processes that fall into the category of epigenetics, explain why not all skin cells of a thorny dragon produce spikes.